This is the walkthrough for the genome comparison. So we're going to do some pairwise genome comparisons here, and we're going to use BLAST and MUMMA to compare them. This is session 4 in the first workshop. So if you go to that first workshop directory, you should be able to open that HTML file. I'm going to be using xming as a terminal, and that's where we want to go now. So let's head off to xming. There we go, there's my xming, and I'm going to take you through from the terminal step by step. So far what we've been doing, we've been working in the BS32010 directory, so I'm just going to move back to that with cd BS32010, and we were in the Pritchard subdirectory and in workshop, and I just hit tab there to autocomplete 1. So if I do ls, that lists what's in the current directory, and I've done a couple of bits already here working through things, but I'll show you what those are anyway. The important file to look at is going to be this one here. This is where our walkthrough script is, and we can open that up just by browsing to it in a web browser. Right, the first thing we need to do, let's take a look at that genome data we've got. So ls genome data, and we can see we've got a number of different organisms there. So let's repeat that command. Up arrow brings back the previous command, and have a look at the E. coli directory. And we've got two organisms there, two sequences in FASTA format. We're going to take a look at that FASTA format, and to do that we'll use the head command. Just use -n 3, I think, will give us the first 3 lines of each of those files. That shouldn't be too bad to see on the screen. So genome data, E. coli, and star to be both files. And what we can see here is that the first line, let me just highlight that, starts with a greater than. Then we've got this identifier, which is a long sort of alphabet soup of different characters and bits and pieces. This is going to be important later. If you download your own file for doing this, download a nucleic acid file for the genome, you're going to want to make a note of this identifier. You will need that later for some of the commands, and I'll tell you when you can replace the identifier that we use in the commands with the one that you will find in your own file. And then there's a description. Here we can see this is the Sakai genome, the chromosome from the complete genome, and this is a different variant of pathogenic bacteria. This one gives you a urinary tract infection, rather pleasant chap, not quite as unpleasant as Sakai there. Okay, so we've got some data, and we're going to need to make a directory in which to put the output. So we can use the mkdir command for that. Now, using -p will mean it won't give an error if that directory already exists. So session 04 underscore output. It could be any name, but remember to keep using that same name that you use all the way through, changing it where necessary in the subsequent commands just to make sure that it all works. Now, you heard in Dr. Pritchard's lecture about using BLAST, and if we use BLAST to compare the big sequences, it will use the MegaBLAST algorithm, and that should probably be pretty quick. Now, if you're looking at the script, you'll see that you can run BLAST n-h to see what kind of options that takes. So let's try that. BLAST n-h, oops, and that gives us a huge list of different options. We don't need all of these by a long shot. In fact, we need very few. This is really for ultimate fine-tuning of the BLAST algorithm, and we're not going to go into that today. Instead, we're going to be using the commands that are written in the schedule. Let's go back to the browser. Okay, let's just hide Xming, and you can see on the browser we've got this long command here. Now this should all be written on one line, and when you write it on one line, don't put in these bits, the backslash at the end of the line. That's just a convention that says skip the new line, ignore it, and just treat it all as if it's on one line. So let's write that lot in. Here we go. We're going to use the time command just to find out how long it takes to run. So time blast n minus query genome data E. coli NC002695 as our subject, as our query. Now I was using the tab key to autocomplete to make typing a bit quicker, and I just need to type the first few characters. As long as it's unique, I can hit tab and it will then behave. Now we're going to look at the output format, output format 6, and that will give us a table. Can provide you with all the details for what is in that table in a moment. I'll find that for you in just a moment. And then we're going to run minus out, and we'll put it in that directory we've just created, and we'll call it E. coli megablast.tab. Okay, so we'll run that, and that should run pretty quickly. Just takes a few seconds, and time will tell us that it only takes about 4 seconds. So that's fine. Let's take a look at what that gives us. Okay, let's head again. Just take the first 5 lines, and we will look at session output E. coli megablast.tab, and this is giving us a table. There are different columns here. I'll show you what those are in just a moment. I think there's something like 12 different columns. These are obviously the query, and this first one here is the query. Then we've got the subject identifiers, the percentage identity, and various other parts in here. I'll show you what those are in just a moment. Bit score, number of gaps, starting coordinate and ending coordinate for each e-value, and so on. So I'll show you those in just a moment, but in the meantime let's compare that to the old blast n, and we can do that quite simply. Let's pull back the previous command, this one. It's rather long. Let's change the output file. Let's call it blast n.tab, not .tab, and then we'll tell it that the task we want it to do is blast n, and we'll let that get on with it. It will take a little bit longer, and there'll be a few warnings there. In the meantime, I will try and find you that nice table of different things for you to take a look at. Okay, so this will be a table containing all those different bits and pieces. I've just put this into a Jupyter notebook that I should be able to add into this browser. Let's take a look there, turn that on, hide Mama for the moment, and we can see here we have the various columns. So the query sequence ID, the subject sequence ID, percentage of identical matches, alignment length, number of mismatches, the number of gap openings, so really the number of gaps, query start, query end, and we end up with e value and bit score, the overall scoring. So 12 different columns, and those are going to be read when we do the comparison to be able to line up all the bits and colour them appropriately. Oh, now BLAST_N is finished. Let's go back to that and see what that looks like. Here we go, and we can see there it's taken about 50 seconds, just under a minute, to run there. Now there is another algorithm, there's one called MUMMA. Now that runs in a slightly different way, and what we're going to do with that is to identify matching regions with it. That will produce a delta file. We're going to get rid of all the tiny matching ones, so from the delta file that will create a filter file, and then we will use show coords to generate a human readable alignment from there. We can plot that as well using MUMMA plot. So we'll go through and do all of these, and then we will visualize it. And then we will move on and use the Artemis tools. So what we want to do is to run NUCMA. Let's see how long this takes. So time NUCMA, and we're going to set the maximum gap. Note here that we have two minus signs for each of the options. So max gap is 500. That's the biggest gap you can have between matching regions and still cluster them together as one cluster. And we will have a minimum cluster size as well. Minimum cluster, the minimum length of a cluster of matches. Okay, so this is the smallest region we're going to take. And once again, if you're looking at the script, we ignore the backslash on the end of the line because we're going to put this all on one line. Prefix tells it where the working directory is and the kind of base name for the file. So we're going to put it into our session output directory, and we're going to call it E. coli NUCMA as our base, and then we're going to read into that from genome data E. coli NC002695 as our query, and likewise E. coli NC004431 as our subject, and we'll hit go for that. And this again is actually not too bad in terms of efficiency. It will read the files, it'll spew out a lot of input or output, and finish up pretty quickly. So let's take a look at what that's actually produced. It's going to produce a delta file, head, session 04 output, and we're going to look at E. coli NUCMA.delta. And this is not the most friendly of files, so it's running down from here. It starts with NUCMA, and then it's really just a lot of miscellaneous numbers. It's quite hard to make any kind of sense of this unless you are the program. We could have a little look at a little more of that. Let's use more to go through page by page. Yeah, this is not really terribly human readable, is it? A lot of numbers, but you really need to know the algorithm and the structure to be able to read that. Okay, Q to quit from more. Let's get this into a more tabular format, more like the BLAST one. Okay, we're going to use a command called show -coords, and we're going to read in session output E. coli NUCMA.delta and put that into file E. coli NUCMA.coords. Okay, note that back here this is taking a greater than symbol. If we just run this command, it will read the data in and then spit it out to the console. So we use the redirect here to take that output and put it into this file. Okay, here we go. Let's take a look at that. We'll use more again. And now you see we've got a NUCMA output again, which tells us which files we've looked at here. That's all on the first line, then NUCMA to compare, and then it's telling us the start in sequence 1, end in sequence 1, start in sequence 2, end in sequence 2, length of the match in sequence 1, length of the match in sequence 2, percent identity, and the tags are the sequence names. So we can recognize this bit here as being the first part up to the first space from the FASTA file, and likewise this one from the other FASTA file. Okay, so we've got a nice tabular output that lets us review that, and if you were feeling keen you could probably drop it into R and do all sorts of interesting things as well with it. Anyhow, we are going to run the show lines to see what we can actually see directly. So run show lines, put in the output file, the delta file, E. coli nucma.delta, and now we need to know the IDs of our sequences. So if you've run this for different sequences, you need to substitute here for the appropriate sequence identifier that you've read out of the FASTA file. I'm just going to take this. The pipe symbol is at the bottom left of the keyboard, just above the backslash key. So shift backslash gives you pipe, the vertical line. And you have to be quite careful typing this to get it exact. 158.29254 pipe ref pipe nc underscore 002695.1 pipe close quotes space, and then I can do the other identifier as well. Four 45917 pipe ref pipe nc 00 underscore 4431.1 close quotes. Right, that's going to throw the output straight onto the console, so I will redirect with a greater than and put it into session E. coli nucma dot aligns. It doesn't like me there. I probably have a typo in there somewhere. Let me just check that. Oh, I've forgotten to put the pipe on the end of the last one there. Let's change that. Still doesn't like me. What have I missed? 26249145917. Ah, somehow I've ended up with an underscore in the wrong place there. That's better, that's now worked. So it is rather fussy. You have to look very carefully and make sure you type things exactly right. Let's go back and find head, or more will do, and look at aligns, and that now gives us the alignments and highlights the variations for each of the sections. So we can see here there's a difference, an insertion in one compared to the other. And we can work our way through and see lots and lots of sequence alignments, pairwise alignments. Right, so far we have been looking at the raw alignment data, and actually what we probably want to do is to filter that a bit. So remember we talked about reciprocal best blast hits. Well, for this alignment what we want to do is just restrict this to that, so that we can map each position in the query only to its best match in the reference, and match each position in the reference only to its best match in the query. So for that we'll use the delta filter command. This will filter a delta file, and we'll do the -q to match the query only to the best matching position in the subject, and conversely the -r to match each position in the reference only to the best match in the query. So in NUCMA they call the database sequence the reference and the search sequence the query. In BLAST the search sequence is also called the query, but the database sequence is called the subject. So let's put in a delta file here, session output E. coli nucma.delta, and again redirect the output because it will spit it on the screen otherwise. Session output to E. coli nucma.filter. Okay, let's just take a quick look at that. We can actually make that more readable. We can use show coords again, and this time instead of delta I'm going to change the delta to filter, and I'm going to change this to nucma_filtered. Has this affected the number of matches it shows? Well, probably yes. Let's do a WC-L. That will count the number of lines in a file. Session output, and we're going to do star.coords, and we can see that we've gone from 721, there's one I did earlier, 721 lines in nucma.coords to 493 in the filtered results. We've got rid of some low scoring rubbish. We are going to now create a plot of that. It's all very well having graphics, having numbers, but a graphical representation gives us a really quick visualization of what the sequence looks like. So let us go and find mumma plot. Here we are, mumma plot, and mumma plot will let us choose various different outputs. There's PDF, there is PNG. So PDF will give us a vector graphics file that we can import into almost anything suitable, such as Illustrator, anything that deals with PDFs really, Inkscape. More useful for presentations and reports could be a PNG format, so we'll use that. This is a graphics format that gives us a bitmap graphic that we can then use in PowerPoint, on Word documents and so on, or even on the web. I'll show you that on the web in just a moment. So we'll take our filtered data, and we're going to have our reference sequence is going to be the NC02, and our query sequence is genome data E. coli NC004431, and our working directory in which to put everything. Again, it's our session directory, and prefix For the file name, it's going to be E. coli nucma. Now we do get a few outputs here. It's using a package called GNUplot, and it couldn't. Don't worry about this error here. It'll just give us an ugly font for the moment. At some point, I'll update the fonts on the system and get that to work properly. But it's drawn us this nice file here, and I've got one I prepared earlier. I've dropped it into a web browser. Actually, what I've done is I've created an IPython notebook with it in. So let me just flick back to the web browser. Here we are, and we can see the comparison. We can see that most of this is really nice. There's something that looks like repeat sequences here, these odd dots, but most of it is pretty much aligned as a nice sequence. There's a few little gaps and bumps and so on, and some repetitive bits. Okay, so we have gone through getting our genomes compared. You can probably do that with your own sequence now. Remember to download it to an appropriate directory and name it appropriately, and take a note of the ID in the FASTA header. What we're going to do next in the next video is to use Artemis comparison tool to go and compare them with a nice sort of, well, nice, it's a little bit clunky, but a graphical view. So if you go off to your browser, what I've done, I've actually copied the whole of my session directory onto my local computer. I've done that with WinSCP. You could do this. The files aren't very big, so you could probably put that onto your H drive on the university laptop, and then we can run Artemis comparison tool. Here's the website. Let me just zoom in a bit on that, and you can launch this much the same way you launch JALView, with a web start. So if you click on launch, that will download a JNLP file. It'll prompt you whether you want to save it or not, telling you it'll do all sorts of nasty things to your computer. Just click yeah, you'll keep that, and then click it to launch. It will then launch Artemis, and follow along in the next video to see how to use Artemis comparison tool.